Assalamualaikum and a very good day I bid to the honorable judges and audiences. First and foremost, I would like to quickly introduce our group members. My name is Afrina and along with me is my partner Alifah. Today, we'd like to share with you a presentation of our project titled The Total Salivary Protein, Flow Rate and DMFT Index Assessment in Year 1 UiTM Dental Students. This is the outline of our presentation. The background of our project will be touching regarding dental caries, salivary total protein, and flow rate. According to the World Health Organization in 2020, there are 2.3 billion people that suffer from dental caries of permanent teeth. Dental caries is a process where dissolution of crystalline mineral structure of a tooth is broken down by acids derived from bacteria. The main causative agents are bacteria, diet, time, and host factors, which include an individual's salivary composition and others. Salivary protein play a major role in protecting oral tissues against bacterial, viral, or fungal infections as well as of protecting our teeth. A study revealed that there is a correlation between salivary flow rate and protein concentration. Consequently, the increase in salivary flow rate will result in an increase of protein concentrations. Due to the diversity in composition and function, saliva can be used for the diagnosis of dental caries. Worldwide, caries is measured with DMF or Decayed, Missing and Filled Index. The scoring is based on the results of clinical examinations and calculation of the number of DMF teeth, ranging between 0 to 28 in permanent teeth. And in our study, we decided to do a further exploration about the correlations between the dental caries and the salivary parameters among the first-year dental students. Hence, the objective of our project are to evaluate the DMFT index, salivary total protein and flow rate in stimulated and unstimulated saliva, to correlate the DMFT index with salivary total protein and salivary flow rate, and lastly, to determine the association of unstimulated saliva with stimulated saliva towards risk of dental caries in UiTM dental students.
Now, let's continue with the methodology part. For the protein analysis, we collected 3 to 4 mL of the stimulated and unstimulated saliva. Later in the lab, we performed biciconic acid protein assay to measure the salivary total proteins in both of the saliva. All our samples were prepared in duplicates and the absorbance of samples were measured at 562 nanometer wavelength. And last, we plotted a standard curve. All data were analyzed using Fisher's exact test, t-test, and Pearson's correlation test.
Moving on to our results and discussion. The study subjects consisted of 57 subjects, where 25% of them are male and 75% are female. Subjects were divided into 4 groups of DMFT, which are Group A with DMFT 0, Group B DMFT 1 to 3, Group C DMFT 4 to 10, and Group D DMFT more than 10. Majority of the subjects are demonstrating the DMFT category in Group B with 40.6% and C 36.9% as shown in Figure 1. The subject were also divided into 2 groups of normal and high protein concentration level in Figure 2, where 89.47% and 98.25% of subjects showed normal concentration in unstimulated and stimulated saliva respectively. For flow rate, the subjects are divided into 3 groups of very low, low, and normal flow rate as shown in Figure 3, where 70.18% and 78.95% showed normal salivary flow rate in unstimulated and stimulated saliva respectively. Answering Objective 2, Table 1 shows the correlation between the MFT and the salivary parameters evaluated using the Pearson's correlation test, where most p-values showed insignificant values. Hence, it cannot be concluded that a significant difference existed between the parameters among the subjects, except for the correlation between protein concentration and flow rate in unstimulated saliva, showing significant negative correlation between the variable with p-values of 0.017 and correlation coefficient of negative 0.314. Answering our last objective, the association between the MFT groups and salivary total protein in both types of saliva were evaluated with Fisher's exact test, as shown in Table 2 and 3, where both tests showed insignificant p-values. Thus, there is no significant association shown between the variables among the first year.
According to the results obtained, we noted that the frequency of DMFT demonstrated that a large number of subjects were carries risk groups, showing 40.6% of the total subjects were in DMFT Group B and 36.9% in DMFT Group C. However, majority of the subjects had normal total salivary protein in both unstimulated and stimulated saliva, which are 89.47% and 98.25% for unstimulated and stimulated total proteins respectively. Based on our results, there is a significant p-value between the protein concentration and salivary flow rate in unstimulated saliva, but it showed a negative correlation, meaning that if the salivary flow rate increased, the protein concentration will be decreased and vice versa. In contrast, study done by Al-Alimi et al. In 2014, they found that increase in salivary flow rate will result in an increase of protein concentrations and electrolytes, so they should have a parallel increment. Theoretically, they also stated that the results may be due to the physiological and histological changes that altered the salivary flow rate throughout lifetime. Corresponding to another study done by Hamadi et al. In 2017, they identified that high levels of total salivary proteins were found in the carries-free group compared to the DMFT group. So from our data, we observed that there was no correlation between the DMFT, total salivary proteins, and salivary flow rate for both stimulated and unstimulated saliva. The association between the DMFT groups and salivary flow rate shown in Table 4 and 5 were also evaluated with Fisher's exact tests, where all the tests showed p-values higher than 0.05, showing no significant association between those two. With regards to the salivary flow rate in unstimulated and stimulated saliva, majority of carries-free group had normal salivary flow rate. As supported by Al-Alimi et al. In 2014, this could be due to normal salivary flow rate offers a strong protection against dental caries. Concurrently, majority of caries risk group also had normal salivary flow rate, and this may be because we measured the salivary flow rate at only one time. And it was also proven by Al-Alimi in 2014 that salivary flow rate varied from time to time. As a result, we found that there was no significant difference in the DMFT for salivary flow rate in stimulated as well as in unstimulated saliva. Principally, our current study focuses on the correlations of total salivary proteins and salivary flow rate of stimulated and unstimulated saliva with the DMFT index of the first year UiTM dental students with comparison to other studies. We noticed that there was no significant correlation between every parameter tested. This is similar to a previous study done by Hamadi et al. In 2017, specified that there was no stable association between salivary proteins and dental caries with regards to the protein phenotypes, protein molecular weight, or total proteins concentrations. Generally, the relationship between total salivary protein and the DMFT index has been studied by several researchers before, but the results are varied. It is notable that many factors could affect the differentiation between each of the study, such as the sample size, the age, gender, and type of saliva as mentioned by Heah et al. In 2019. The limitation of our study is a narrow distribution of age and gender, which only involve subjects ranging from 19 to 21 years old. To conclude, majority of first-year UiTM dental students had normal salivary protein concentration and salivary flow rate in unstimulated as well as stimulated saliva. However, with no significant correlation between these parameters with caries incidence. For future study, we recommend to increase the sample size and range of age of the subjects for better results. So these are our references. With that, thank you.